Okay, can somebody confirm you can hear me?
I can hear you.
Yes.
Great.
Yes.
Sorry for being late. There's about 8 clicks I have to do to get the class running, and my computer is a little slow. Let me get going here. Before we begin, are there any questions? All right, as I stated, my computer's a little slow. I'm trying to get the lesson up. All right, here we go. Let me share my screen. Should be seeing my screen now. That didn't go to the last slide we were at. My computer isn't working right. There we go. All right, so we had talked about the astounding numbers that bacteria were at. This is the last slide we had talked about. And let me briefly show my syllabus. Boy, that's the wrong color. Something isn't working right on my computer. Today, we're going to be continuing chapter 1 and chapter 10. We'll have a lab today, and that'll be lab 01. The lab will be at the normal time, which is, I think, 6:30. Any question about any of that? All right. So we've talked a little bit about microbes and we've talked about disease. Well, microorganisms can cause disease. We call them pathogenic if they are a microbe that can cause disease. So a pathogenic microbe is a microbe that causes disease. Now understand that most microorganisms do not cause disease. Only a small percentage are pathogenic. Only a small percentage of all microbes do cause disease. When we're talking about disease, it's important to distinguish between infectious diseases and an inherited disease. And an infectious disease is caused by a microbe or a parasitic worm. What's an example of an infectious disease? Can anyone name an example? An infectious disease? Well, think about that. Can anyone name an example of an inherited disease? An inherited disease is one that you get when you are born. So you get an inherited disease from your genetic makeup. And nobody's going to give any examples. Sorry, can you say that again?
Sickle cell?
Yeah, sickle cell anemia is an excellent example of an inherited disease. You only
Diabetes type 1.
That is not such a good example because it does have a genetic component, but it does have an environmental component, meaning someone who isn't taking care of themselves and isn't eating appropriately, they eat a lot of sugar or something like that, they're much more likely to become a diabetic than somebody who's taking care of themselves and then they avoid a high carbohydrate or high sugar diet. So
Wouldn't that be diabetes type 2?
no, like I said, both types of diabetics, there is a genetic component to it, but there's an environmental component as well. So like I said, that's not a good example. That's like somebody's weight. If they're overweight, they might have the genes that help them to be overweight, but it depends on how much they eat that will really determine how much overweight they are. So that is not a good example of an inherited disease. An inherited disease is one where you get it when you're born because of your genetic makeup. And an example of that would be, well, obviously sickle cell anemia is a good example, but I'm trying to remember. Tay-Sachs disease would be a good example. There are a bunch of other examples. I don't know if anyone would know of them, like Xernoderma pigmentosum. I don't know if anyone knows what that is, but and what is a cystic fibrosis is another example. An infectious disease is one caused by a microbe, and that could be something like the human cold, the human flu, COVID-19. Food poisoning, assuming they're getting something in food. Trying to think of another case, botulism, which, although it is infectious because you can get it from eating something with the botulism toxin or the botulism organism, that disease is unlikely to spread from one patient to another. Whereas something like the cold and the flu are easily capable of spreading from one patient to another. But all the infectious diseases are caused by a microbe. Any question about the difference here? All right. So we talked about some microorganisms. They are small. How small are they? This picture is showing you. Let me see if I can blow this up a little. This looks like it could be E. coli. It's a little bit bigger, longer than most bacteria. It is a little bit less than 1 micrometer on its diameter or its length. No, the width, sorry, the width. But on its length, it's a little bit longer than most bacteria. Most bacteria are on the size of around 1 micrometer, maybe up to 2 micrometers. And that's about your average bacteria. This is a fungi, and this is probably mold, and this would be an individual cell. Sorry, my mouse doesn't work. There it is. That would be an individual cell here. That would be an individual cell there. And you can see that's quite a bit larger. I can barely read that. 10 micrometers long right there. This is a microbe, but this is actually a polyclonal microbe. Each green dot here is a cell. So this is polyclonal, but it is microscopic. You can't really see it with your human eyes. And this is actually, these right here are 5, make that 6 baby colonies inside the parent colony. And each of these dots there is a cell. That's actually a white blood cell. It's also microscopic. And that's about the largest. We've got about 50 micrometers long. And so that's what, maybe 200 micrometers in diameter. And then this is showing you a T cell with each of these red dots being an HIV virus. So they're small microbes. They come in various shapes and sizes. Microbes play many beneficial roles in both the ecosystem and for man. For example, we already mentioned microbes decompose organic waste. They're involved in the nitrogen cycle. They're largely what makes the nitrogen cycle work. They are producers in the ecosystem. A producer is something that can fix carbon, like in carbon dioxide, and then make glucose out of it. And you all know photosynthetic plants do that, but there are also photosynthetic microbes. And then the chemosynthetic microbes are also producers. They can fix carbon dioxide and make glucose. Microbes produce a large number of different things for people, like industrial chemicals, like we use microbes to make ethanol that you may put on your cars. Since most of you are in Western Washington, we put ethanol in our cars, at least in Western Washington. You know, I don't know about Eastern Washington, probably the same. We use microbes to make acetone. We do use microbes to make some insecticides. We use microbes to make fermented foods, such as vinegar, cheese, bread, beer, and a bunch of other foods like kimchi, sausage, even chocolate. We use microbes to make products made in manufacturing, like cellulase. We use microbes to make antibiotics, like penicillin G is made completely by a microbe. Ampicillin is made by a microbe, but then we chemically modify it to make it ampicillin. And then microbes are used for making other products used in medicine. For example, human insulin is now made in a microbe, and then they purify it, and then they give it to the diabetic patient. Not sure what happened there. I was supposed to move on to the next slide. Hopefully we're still, no, I'm not even sharing the screen here. Why isn't that advancing? There we go. Hmm. All right, microbes are used in biotechnology and genetic engineering. Biotechnology is the use of microbes and other organisms to produce foods and products for human use. Biotechnology is actually centuries old. Like sauerkraut uses microbes to make it, and that's been around for centuries, maybe even longer. Genetic engineering is a new technique of biotechnology. And so when most people talk about biotechnology, they're actually referring to genetic engineering. There are different applications of genetic engineering where we can use microbes to produce a variety of proteins from either bacteria or fungi. We can use microbes for gene therapy, such as correcting the genes that are defective in a baby that's born. And we might talk a little bit more about that, but like a child that's born with a defective immune system, you can pull out their immune system cells, grow them up in vitro, and then infect them with a microbe. Usually it's a virus, but it could be another microbe, which carries the correct gene in, and then those cells can be injected back into the patient, and then the patient has a functioning immune system. And then we use microbes to make genetically modified bacteria or genetically modified crops. There used to be the flavor saver tomato that the stores loved because it kept the tomato from spoiling quickly. Normally, the tomato would last much longer, fully ripe on the store shelf. However, I have not seen that tomato in the supermarkets for a long time, so apparently it wasn't very popular with consumers. Let's talk about microbes and human welfare. Microbes live with humans so that humans have a microbial ecology with them. A microbial ecology is where microbes are responsible for doing something in the ecosystem. We already mentioned that bacteria can recycle nitrogen and cause the nitrogen cycle to function. Well, bacteria also recycle carbon and help the carbon cycle recycle on the planet Earth. And the same thing with nutrients, the sulfur cycle and the phosphorus cycle. All of these cycles have microbes which help the cycle work. The best would be the nitrogen cycle, where almost every step in the nitrogen cycle is happening because of a microbe. In the sulfur cycle and the phosphorus cycle, microbes are important, but there are some steps like the leaching of the minerals from rocks, which usually happen just by water. Microbes aren't important in getting the first step of the cycle going, but they are important for converting one form of the cycle to another form of the cycle, meaning one chemical element of the cycle, moving it to another chemical element in the cycle, keeping the cycle moving. Nutrients are recycled by microbes. So when somebody dies or some organism dies, the microbes will make those nutrients available to another organism, usually a green plant or something like that. These are all examples of microbial mediated biochemical cycles that support life on Earth. If we didn't have the microbes, then all of the chemicals in all of these cycles, carbon, nitrogen, nutrients, sulfur, they would all stop at one point in the cycle. And then they wouldn't move from that one chemical to the next chemical in the cycle. And then the cycle would essentially stop. And that would be detrimental to life on Earth because we need these elements and nutrients recycling on the planet Earth for life to continue. The microbial mediated biochemical cycles were first discovered by Winogradsky and Bajering in the 1880s to 1892. And they first discovered this, I think they first discovered it on the sulfur. And I used to remember this. Is it the nitrogen? I think they first discovered it in the sulfur and the nitrogen cycle. And we'll talk a little bit more about that a little bit later, what a Winogradsky column is and why they were able to discover this. What's going on with a bunch of people adding the class and dropping the class? Nobody knows? All right. Microbes are also important in bioremediation. Bioremediation is important for human welfare because when there's a toxic spill, it's the microbes that generally break down those toxic substances. Like what these men are doing is there was an oil spill here and they're spraying microbes on the oil to break down the oil. Bioremediation literally means life into remedy. And of course, we use microbes to break down sewage, and everybody knows that because we've already talked about it. Boy, my screen is not working well. Can somebody confirm? Oh, there we go. You can't see it. We also use microbes to break down mercury, which doesn't really remove the mercury, but it changes it from one form to another, making it a less toxic form of mercury. And we use bacteria to degrade and detoxify pollutants as well as waste products in sewage and break them down to less harmful substances. Like oil, as well as everything in the sewage, will be broken down to water and carbon dioxide. Mercury will be converted from elemental mercury to a organic form of mercury, which is less toxic to us. Microbes are also used for making biological insecticides. So some microbes are pathogenic to insecticides, and these are alternatives to chemical pesticides. Chemical pesticides are generally toxic to people. That is not the case with microbial insecticides. And then we can use these microbes or microbial insecticides to prevent damage to crops, as well as to prevent disease transmission. Usually that's crop disease transmission. An example is Bacillus thuringiensis. This bacteria is actually fatal to many insects. And yet this bacteria is harmless to humans and other animals, as well as to plants. And this is an example of Bacillus thuringiensis, which my father actually bought in the 1960s, and he put it on the apples of his apple trees. However, he only put it on one time, and you do need to put on Bacillus thuringiensis about every 2 weeks, which is the same as using the chemical pesticides. So it did decrease the worms in the apples, but he was far too lazy, and you need to put it on, like I said, every 2 weeks. And also he put it on the apples in Western Oregon. And as you know, apples ripen in the fall in Western Oregon or in the early winter. And it's pretty wet in Western Oregon at that time of the year. And after the rain will just wash Bacillus thuringiensis off the apples. So you need to apply it after every rainstorm and also after every 2 weeks. And if you do that, you will get your apples without worms, or pretty much without worms. And it is used like organic apples when you buy them in the store. Any question about any of that? All right. Let's move on to the naming and the classifying of microorganisms. Both the naming and the first classification of microbes, as well as all other organisms, began by this person here. Does anybody know who started the scientific nomenclature, as well as the classification of all organisms? You should have had that in Biology 160. Nobody knows? Well, think about it. Each organism has 2 names. If it's a scientific name, it has 2 names, why we call it the scientific nomenclature. And that's why it's also called the binomial nomenclature. Binomial means 2 names. So a species scientific name includes both the genus and the specific epithet. So Homo sapiens is the name of the species for humans, Homo being the genus, and the specific epithet being sapiens. Any question about any of that? So when you're talking about the species name, you cannot just say sapiens. You must say Homo sapiens. Anyone know who this is? Anyone remember from Biology 160?
I don't know how to say his name, but is it Carolus Linnaeus?
Yeah, that's almost exactly correct. Carolus Linnaeus, and he liked Latin so much, he Latinized his own name. His real name, and he's from Scandinavia, I think he's Sweden, was something like Carl Linné. I might be not pronouncing that correct because I'm not Swedish, but he Latinized it to Carlus Linnaeus. And it's curious, your textbook calls him Carl Linnaeus, meaning they Latinized his last name, but not his first name. But either way, either name is correct. And he's the guy who started the scientific nomenclature, a system which was so good and so widely used that it's still used today. So he gave humans the name Homo sapien, which literally means wise man who walks on 2 feet or something like that. He also started the classification of organisms, and his classification scheme is no longer used because he classified all organisms into plants and animals. And as you know, a microbe is not really a plant or an animal. And that's why we no longer use his classification system, as well as fungi and a few other organisms do not fall easily into a plant or an animal. We'll talk a little bit about the classification system, but we're going to talk a little bit more about the scientific nomenclature first. So when we talk about a scientific name, the name should either be italicized, or if you can't use italicized, you underline it. The genus name is capitalized, and the specific epithet is lowercase. So Homo is capitalized, sapiens is lowercase. And that is in italics, because I can do that typing. But like when I'm writing on the board, I'll write Homo sapiens and then put an underline underneath it to make it clear that that is a scientific name. All scientific names are Latinized and used worldwide, meaning wherever you are in the world, you use the same scientific name, regardless of the language you speak. So Homo sapien always means humans. The name may be descriptive or it may honor a scientist. So Homo sapien is a descriptive name. Like I said, sapien means wise man, and Homo means man who walks on 2 feet or species that walks on 2 feet. Escherichia coli is descriptive and honoring a scientific name. Dr. Escher was an early microbiologist, and Escherichia was named after him. So that name is honoring the scientist. Coli is descriptive because Escherichia coli can be found in the colon. Staphylococcus aureus is descriptive. This literally means golden sphere that grows in a cluster like grapes. So aureus means golden, coccus means sphere, and then staphylococcus means cocci that grow in a cluster like grapes. Any question about any of that? When using the scientific nomenclature, first use, you should spell out the long name. But after first use, you may then abbreviate the scientific name with the first letter of the genus, followed by a period, and then write out the specific epithet. So Staphylococcus aureus should be used, spelled like that the first time you use it, but after that, you may abbreviate it S. aureus. E. coli is supposed to be written out the first time now I can't say it, Escherichia coli, and then all other times E. coli. However, E. coli is an exception that even among scientists, they will frequently use E. coli the first time they use it. And that's because E. coli is better known than Escherichia coli. That's about the only exception. Any question there? All right, let's talk on to the classifying of microorganisms. As I stated, Carolus Linnaeus initially classified all organisms into plants and animals. That system is no longer used and has not been used for at least about 100 years. We now classify all living things into a 3-domain system. Also called the tree of life because the branches of the tree of life branch much like a tree, although this is a tree with 3 trunks, but each trunk looks like a tree. And if you have a tree that has 3 trunks, it's possible, then that is a tree of life. The domains are based on the genetic makeup of the species within the domain, meaning if we were to sequence the domain of a bacteria, its sequence of DNA will be more similar to any other member of this domain bacteria than any member of the other 2 domains, Archaea and Eukarya. Is that clear? Anyone confused about that?
Wait, can you repeat that? Sorry.
The domain is based on genetic data, meaning any organism within a domain will have its sequence of DNA more similar to any other member of that domain than any member that is outside the domain, meaning the other 2 domains. So if we ever find another organism on the planet Earth, all you need to do is sequence the DNA of that organism, and then that will tell you immediately which domain the organism falls into.
Okay, thank you.
And there are 3 domains: domain bacteria, domain archaea, and domain eukarya. So there's no disagreement about what a domain is and where an organism falls into which domain, because we just sequence the DNA and it tells us what domain the organism belongs to. Any questions about that? That's different from a kingdom and essentially all other groups underneath a domain. The number of kingdoms there are is actually in disagreement. Some people say there's 3 kingdoms, other people say there's 5 kingdoms. Most people say there's 6 kingdoms, so we'll use the 6 kingdom approach. But some people say there's 9 kingdoms. Some people say 12 or 15 kingdoms. Some people say 20 different kingdoms. And then other people higher orders of kingdoms above 20. And I don't know the number above 20, but there's at least a 20 kingdom approach, as well as higher. The reason why there's a disagreement and why we don't say that there's one kingdom approach that the majority of scientists use is because there is no definition for a kingdom. We will use the 6 kingdom approach, and that has the kingdom bacteria, which is the same as the domain bacteria, the kingdom archaea, which is the same as the domain archaea, and then the domain eukarya, which is split into 4 different kingdoms. the kingdom Plantae, the plants, the kingdom Animalia, the animals, the kingdom Fungi, the fungus, and then the kingdom Protista, the protists. There is a disagreement as to which kingdom an organism falls into, even if you're using the six kingdom approach. Like there's, for example, some kelp, some people consider them to be plants, other people consider them to be protista. There are some microbes which have characteristics like euglena of an animal, but yet it can be photosynthetic, meaning light, can make glucose using light and carbon dioxide. Some people consider it a plant. Most people consider it a protista. Any question about any of that? Now, like I said, there are different kingdom approaches. With the 9 kingdom approach, and I think the 20 kingdom approach as well, all you do is you have the same kingdoms. You just split this kingdom Protista into many different kingdoms. And actually the Protista, when we simply have the 6 kingdom approach, the Protista is defined as a eukaryote that is not a plant, not an animal, not a fungi, which means that the Protista is a collection of other organisms that are not genetically related to each other, meaning they're not from one phylogenetic, they don't have a phylogenetic common ancestor. So we've always known the Protista are a collection and you can split it into more than one group. And certainly the 9 and 15 kingdom approach do that. I think the 20 kingdom approach does also, although the 20 kingdom approach may split the plants into 2 kingdoms. I don't remember. Any question about any of this? All right, we won't talk about the lower classification system, but do realize after kingdom, let's see, you have subkingdom. After subkingdom, you have a class, then you have order, family, genus, if I got it right, and then species. We won't talk about any of those others, but you do need to know that all living things can be divided into the 3-domain system. The 3-domain system can be divided into the kingdom system, and we're just going to use the 6 kingdom approach, which is the approach that the plurality of biologists use, but not all biologists use it. It is the last kingdom approach that the majority of biologists used to use, but when we started sequencing organisms, we realized that the 6 kingdom approach is not a good approach because the kingdom Protista has a lot of different organism in it. So that really should be more than one kingdom. All right, your textbook talks about a cell type and says there are 2 cell types, prokaryotes and eukaryotes. Bacteria and archaea have the cell type prokaryotes, and then eukarya have the cell type eukaryotes. Some people, not your textbook author, call this a super kingdom. And a super kingdom was actually around long before the domain came about. The domains never came about until people started sequencing species. So the domains came about, I think, in the 1970s. The super kingdom approach came about at least in the 1960s, if not the 1950s, meaning we knew about prokaryotic cells and eukaryotic cells long before we knew about the domain. And that's why we call it a super kingdom. However, your textbook doesn't use that term. It uses the term cell type. A prokaryote cell type is a cell that has no nucleus and no membrane-bound organelle. A eukaryote cell type is a cell that has a nucleus and membrane-bound organelles. Any question about any of that? All right, let's talk a little bit more about the domain bacteria. They have the cell type prokaryotes. They also have peptidoglycan in their cell walls. So all bacteria that have a cell wall has this chemical peptidoglycan. The domain bacteria multiply mostly by binary fission. That's where one cell will divide and split into two. It splits evenly into two, why we call it binary fission. For energy, bacteria can get their energy needs from an organic chemical, and that's the same as us. We largely get our energy needs from glucose, and most bacteria do as well. But some bacteria can use an organic chemical to get their energy needs, such as from iron or sulfate, or maybe even hydrogen sulfide gas. And then some bacteria can get their energy needs from photosynthesis, getting their energy needs directly from the energy in sunlight. And you all know green plants can do that. Most of the members of the domain bacteria are either primary producers or decomposers. But some members of domain bacteria are parasitic, and some of these parasitic are pathogenic, meaning a few of them are bacteria that can cause disease in people. Any question about any of that? If not, let's move on to the domain archaea. They have the cell type prokaryotic. Does anybody know, do the domain archaea have peptidoglycan in them?
No.
That's correct. Archaea do not have peptidoglycan in them. The only domain that have peptidoglycan is the domain bacteria. Some of the members of archaea are found in extreme environments, and that is where we first found the archaea. However, when we looked further, we found archaea, or members of the domain archaea, in all of the Earth's biomes. So you can find them wherever you look. The extreme environments include the methanogens, organisms that live in a methane and make methane environment. Extreme halophiles, that's extreme salt concentrations, like the Dead Sea between Palestine and Jordan. Very salty. Halophiles grow in that sea. Extreme thermophile, these are organisms that grow at high temperatures. But like I said, we can find them in all the Earth's biomes. The domain archaea actually make up a significant portion of seaweed in the ocean and a significant portion of the microbes in soil. Does anybody know, or does anyone want to guess, are any members of archaea pathogenic? It's a yes or no question. Nobody wants to guess. This one I need to tell you, so I will supply this answer, and that is no. No members of the domain archaea, at least that we know of, are pathogenic, meaning no members of archaea cause human disease. Because of this, we will not study the archaea much beyond this lesson. And that's because, not that because they're not important, they are important, but This class focus is on the microbes that cause human disease, and the archaea do not cause any human disease. So we're just not going to talk about them very much in this class. Any question about any of that? All right, let's move on to the domain Eukarya. This domain has 4 different kingdoms: the plants, the animals, the fungus, and the protista. All of the cells in the domain Eukarya lack peptidoglycan. So if you ever find an organism that has peptidoglycan, it is a bacteria. The kingdom fungi, that's plural, singular, fungus, tend to have chitin in their cell walls. They use organic chemicals for their energy, like glucose, the same as what we use, organic chemical for our energy. You can have multicellular molds, multicellular mushrooms, which are composed of masses of mycelia, shown here, filaments, like that's a hyphae. But you can have fungi, which are also unicellular, like all the yeasts are unicellular. Most of the fungi are decomposers, but a few of them are pathogens, meaning a few can cause human disease. About the only pathogen I think we'll talk about in this class, which is a member of the kingdom fungi, is Candida albicans, which actually is a normal member of our skin, but it can cause human disease, including skin disease. And so it is a human pathogen. I think I already gave that. What happened here? Let's move on to the kingdom Protista. There are some plant-like protists, we call them algae. Realize that not all algae belong to a microbe, like the kelp that washes up on the Oregon and Washington beaches is not a microbe, but it is an algae. So some algae are microbes and some algae are not microbes. Microscopic, not microscopic. They're macroscopic organisms. They have cellulose in the cell walls, the same as plants. They engage in photosynthesis for making energy. So there's a algae, which you can see is green. They do produce oxygen and organic compounds like glucose. And that's one of the types of protista. The second type of protista are the protozoa, and they are the animal-like protists. They include amoeba, paramecium, euglena. They all absorb or ingest organic chemicals. They may be motile, like amoeba, paramecium, and euglena are all motile. And they may be motile by either pseudopods, shown here. So this organism puts down the pseudopod here and then pulls up the cytoplasm here and then moves that way by pseudopods. And that's a white blood cell, by the way. But some of them are motile by cilia. Cilia are microscopic hairs. I'll show you some cilia a little bit later. And then some of the protozoa are motile by flagella. Now remember, if ever we're talking about a bacteria or archaea, if they're motile, they're only motile by flagella. That's the only way that bacteria or archaea can be motile. They are not motile by cilia or pseudopods. Any question about any of that? The protozoa, some of them are parasites, and some of them can cause disease in humans. Like euglena does not cause disease in humans, and it's not parasitic either. But Entomoeba histolytica, an amoeba, is an organism which can cause diarrhea in humans. And then the third group of the protista are the fungus-like protists. We're not going to talk much about them, but these protists are like fungi. And the only one I'm going to mention is water mold. It's a fungus-like protist. And if you don't know what a water mold is, it causes disease in potatoes and actually caused the Irish potato blight, which damaged the main food supply in Ireland, which brought a whole bunch of the Irish over to America, I think in the late 1800s. Any question about any of that? All right, let's talk a little bit about the kingdom Plantae. You can separate the plants into mosses, ferns, conifers, and flowering plants. All members of the kingdom plants are multicellular. We're not going to talk about the kingdom plants any further because none of them cause human disease. Let's move on to the kingdom animals. The animalia may be separated into sponges, the worms, also called helminths, the insects, and then the vertebrates. Humans are vertebrates, if you don't know. All of the members of the kingdom Animalia are multicellular. The worms, or scientific name helminths, can cause disease in people. And so we will talk a little bit about the kingdom animals, particularly about the worms in this class. Worms, at least the adults, for most of them, are multicellular and large enough that you can see them, so they're not a microbe, at least the adult. However, microbiologists do study the worms because the infective stage of all worms is microscopic, usually the fertilized egg, but it could be a microscopic worm that can burrow into your skin, for example. And then they do cause disease, and microbiologists tend to study organisms that cause disease. So the worms are considered a microbe. I don't remember if we mentioned this or not. And it's mainly because they cause disease, and then the infective stage, at least for humans, is microscopic. Any question about any of that? All right, so what comes under the umbrella of microorganism? Obviously, the bacteria and the archaea. Virus and prions also come under a microbe, but understand that these microbes are not organisms. I do not use the term microorganism because they're not an organism. To be an organism, you have to have a cell, and viruses and prions do not have cells. Certain of the eukarya are microbes. All members of protozoa are microbes. All yeasts are microbes. Most molds are microbes. Some algae are microbes. And then the helminths, the worms. The adult isn't microscopic, but the infective stage is. When we're talking about the helminths and say they're multicellular animals that we call worms, you should come to understand that there are actually 3 different worms: the flatworms, the roundworms, and the annelids. A earthworm is an annelid. And when we sequence the DNA of these worms, they shouldn't be lumped together in one group we call a helminth, because the flatworms and the roundworms are as different from each other as they are to humans. And they're as different as humans are to each of these, meaning the flatworm and the roundworm are as different from each other is as a human is different from a roundworm. We call them all worms just because they all look like a worm to us. So it's sort of an ethnocentric term. They should be grouped into 3 different groups. We're only going to talk about 2 of the helminths because only 2 of the helminths are parasitic. The flatworms and the roundworms, also called the platyhelminths, platyhelminthes, and the nematodes. The annelids, the earthworms, are not parasitic. They do not cause human disease, but the flatworms and the roundworms do. All right, and then we call them a microorganism, even though for most of them, the adult is not a microbe, but some of their life stages are microscopic, including the infective stage, which infects the human. Viruses, let's talk just a little bit about them, are a little different from all other microbes because they are acellular. They do not have cells. They do have a genome. However, this genome may be DNA or it may be RNA. It is never both DNA and RNA. If you don't know, all cellular organisms, their genome is always DNA. It's never RNA. The virus has the genome surrounded by a protein coat. The coat may be enclosed in a lipid envelope, but not all viruses have a lipid envelope. And then viruses replicate only when they are in their host cell. When they're outside their host cell, viruses do not replicate. In fact, they have no metabolic activity outside their host cell. Because they do not have cells, they are not part of the 3-domain system. Any question about viruses? All right, this is showing you the different domains of the cellular organisms. Bacteria, archaea, and eukaryotes, giving their cell types. The cell type for bacteria and archaea is prokaryotes. For eutypes, the cell type is eukaryotes. The chromosome for bacteria is generally one circular chromosome, meaning the chromosome is in a circle, meaning the ending point is the same as the starting point. Any question about that? But it differs from the eukaryotes of, a chromosome of a eukaryote, which not do we have one, but we have many chromosomes, and they're not circular, they're linear. Like the X chromosome shown here will have a beginning site and an ending site, and the ending site is not the same as the beginning site. For eukaryotes, we have histone proteins on the DNA. Histones are absent on bacteria. Any question about chromosomes? When we look at the ribosomes of bacteria and archaea are smaller than the ribosomes of eukaryotes. When we look at the cell wall, the bacteria will have peptidoglycan in their cell wall. The archaea have no peptidoglycan, and the eukaryotes have no peptidoglycan. All right, I'll briefly get started on the history of microbiology. When we're talking about a brief history of microbiology, please understand that it largely concerns, at least the early history of microbiology, largely concerned the debate over whether spontaneous generation gave rise to life, or if the theory of biogenesis gave rise to life. The theory of biogenesis states that all life comes from pre-existing life. And then the early age of microbiology, or the early start of the history of microbiology, is called the golden age of microbiology, from about 1857 to 1914, which is the early age of microbiology, where wherever somebody looked, they essentially discovered something new. The golden age of microbiology. This is showing you the history of microbiology, with the golden age being right here. And you can notice that before the golden age of microbiology began, we had some things occurring which were important to the history of microbiology, but they're not literally microbiology, because microbiology hadn't started yet. And then we have some things in the history of microbiology, obviously after the golden age. We'll talk about some of those things. When I'm talking about the history of microbiology, I'm going to give you a date, which is just something to organize my slide. You do not need to know any of the dates except for this very first date. The very first date I'm going to give you is greater than 3.5 billion years ago. We have in the fossil record, meaning in fossils in rocks, the first ancestors of bacteria were seen. And this is the first life that we know of on the planet Earth. Here you're actually looking at a fossil. Come on. And there's the artist's rendition of it. And that looks like a bacteria. It's actually an ancestor of a bacteria. Before that time, we didn't really have any fossils, so there probably was no life. But after 3.5 billion years, we then started getting fossil record of life in the rocks. At 4,300 BC, the Babylonians gave the first written record of microbes. And that is, they wrote on their clay tablets recipes for making beer. And it was the microbes which actually made the beer. That wasn't known to the Babylonians because they couldn't see the microbes, but they did give records for making beer, a recipe, and it obviously allowed the beer to ferment so that it could make the beer. And then the microbes fermented the beer, made the beer. And then after that, in 600 AD, the Mayans made the first fermented beverage from cacao that we would call chocolate. You should know that before the Mayans gave their written records, we have written records of the Babylonians. And then before that, we have in the fossil record ancestors of the first life on Earth. So you should know the relative order of these dates, but you do not need to know the dates. Any question about any of that? All right, the next observation that was important in microbiology was the invention of the microscope, which happened in 1590 by Janssen. He developed the first compound microscope. He actually developed the first microscope, which was a compound microscope. You will note that the first use of a microscope on living things, and anyone reported it, happened in 1665. That's actually 75 years after the invention of the microscope. Robert Hooke took the microscope and looked at plant cells and saw little boxes that he called cells. Let me see if I can demonstrate that. I'm not seeing. Oh, there we go. Paint. So what Robert Hooke did was he looked at shavings from the bark of the cork tree, and in those shavings he saw, that should be straight, what he called cells. And he called them cells because in a monastery, the monks' rooms, so this would be the hallway here, there'd be a door there. The monks' rooms were called cells. And this reminded him of the monastery in the area where the monks' rooms were, which they called cells. And so he called it the same thing, a cell. And if you were to look at the rest of the cork bark, you'd see these cells everywhere in the cork bark. Robert Hooke did look at animal cells, animal tissue, and he did not see any cells. Why was he able to see cells in plants, but not in animals? What do plants have that animals lack? Come on, somebody knows this. What do plants have that animals lack? Think about it. I'm not going to fill in this one.
A cell wall.
Yeah, plants have a cell wall, and that's what Robert Hooke was able to see. The microscopes were not good enough in 1665 to focus in on a cell membrane and see the cell membrane. But they were good enough to see the thicker cell wall. And that's why he was able to see cells in plants, but not in animals. His discovery of cells in 1665 marked the beginning of the cell theory that all living things are composed of cells. That theory was later developed and written up by Virchow, and we'll talk about that a little later. But the first work came from Robert Hooke finding cells in plants. The above are not specifically microbiology, like a plant is not a microbe. And obviously the development of the microscope was not microbiology, but they were important to happen before microbiology could begin. Let's move on to talking about microbiology beginning. It actually began with this guy, Antony van Leeuwenhoek, who was the best microscope builder of his time, and he actually ground his own lenses. And so he was obviously the best lens grinder of his day. He made a very simple microscope here, which was a simple microscope which only used one lens to magnify. So his microscope literally was a super duper magnifying glass. But his microscope was so good, he was able to see things that the compound microscope in his day could not see. He was the first person to observe microbes, and he described their occurrence in almost everywhere he looked. Like he found microorganisms in scraping from his teeth, in rainwater, in peppercorn infusions, and in the water from the River Thames, which was the drinking water for the city of London at the time. Furthermore, using the microscope over here, he was able to see cells so small they were not seen again for literally a century. And what cell he saw are these right here. And as you can see, that's a bacilli, that's a bacilli, that's a spirochete. Figure E, those are conchi. And then figure F, those are bacilli, although that's either two bacilli together or a bent one. So he actually saw bacteria about 100 years before anyone else. And the reason being, well, he made the best microscopes. It wasn't until 100 years after Antony van Leeuwenhoek saw bacteria that the development of the compound microscope got better and got to the point where people could use it to see bacteria. Any question about any of that? All right, let's go back to where we were. Early microbiology was specifically was dealing with two competing hypotheses to explain where life came from. In the 17th to 19th centuries, there were two explanations for where life came from. Some people believe that life spontaneously generated and arose out of pre-existing organic matter or matter. They said that a vital force came into the non-living matter and then spontaneously generated life. The theory of biogenesis states that all living things came from a pre-existing living thing. Now, when we're talking about complex organisms like a baby, there was never any question that babies did not spontaneously generate, that a baby always came from its two parents. Okay, but other organisms, some people believe that they did generate spontaneously. For example, there are things that people used a century ago, like they threw a log into the fire and a newt would crawl out. People would say, spontaneous generation of the newt. And then flies would emerge from manure. People would say, spontaneous generation of the flies. And then maggots arising in a decaying corpse also. Most were logical, but not scientific. Some were even amusing, like somebody wrote up that if you put sweaty underwear in a jar and had wheat in the bottom of the jar, it would spontaneously give rise to mice. In reality, the mice climbed down into the jar to get at the wheat kernels and then got stuck in the jar because they couldn't climb up the jar. And the sweaty underwear had nothing to do with the spontaneous generation of the mice. A brief sidetrack, when we were looking at microbes, you can see a media that has no microbes in it will be clear. You can see right through the media. Whereas a tube of media that has microbes in it will be turbid or cloudy like this. And then when we made media and put it into a tube, the media usually became turbid. And people would say this is because of spontaneous generation of the microbes. The first experiment was conducted by Francesco Redi, and it's also the first documented and controlled scientific experiment. So you should know it for both reasons. He proved that spontaneous generation did not happen for at least larger organisms like flies, maggots and flies. And then it's also the first to write up a controlled scientific experiment. What he did was he got 3 jars, sealed them, 3 jars, covered them with a fine wire net, and then 3 open jars, left them open to the air. In the jars that were left open to the air, the maggots, he put meat in the jar, the maggots appeared in the meat. But in the jars that were completely sealed, there were no maggots. And people initially objected, saying, you sealed the jar. That did not allow the vital force to come in. So that's why you have the jars that were covered with a fine wire mesh netting. Then the vital force could come in. And there were no maggots. So from Francisco Redi's day, everyone agreed that complex organisms did not spontaneously generate. However, there was still debate about whether microbes could spontaneously generate or not. When I go over like this, somebody should let me know, and then I'll stop the lesson. Are there any questions? If not, we will begin the lab. Let's begin 4 minutes late because I'm ending the lecture 4 minutes late. So the lab,
I did have a question.
Uh-huh.
Would you please pronounce your last name for me?
it's Volpe,
Volpe.
Volpe, but Volpe is
Volpe.
close enough.
Okay,
Yeah,
thank
it's
you.
Italian, so the E is pronounced.
Italian. Okay. All right. Thank you so much.
All right, no problem. I'll see you in the lab at 6:34.